Good afternoon, good morning, from wherever you are. Thank you for our online audience for joining us. My name is Julie Ojango. I work at the International Livestock Research Institute based in Nairobi,
Recording in
Kenya.
progress.
Right. And I'll be your moderator for this evening. Now, though we are on different time zones depending on where we are listening into, in Africa, at the close of the day, it is normally the community leaders who gather together to reflect and deliberate on what has been going on, what they have heard, what they have seen during the day, and they use their understanding to guide what they should do going forward. And as was said wisely by a prophet, wisdom and knowledge will be the stability of your times and the strength of your salvation as a people. So, as you've gathered here this evening in the continent of Africa, it's morning, it's afternoon elsewhere, but we want to deliberate on the Africa we want as part of the CTLGH and Livestock Biotechnologies vision, in line with the Africa Union Agenda for 2063. So welcome, and we look forward to a very, very interesting discussion and presentation from different people. We're going to get very many perspectives. Now, a little bit about the event. Food and nutrition security remains a challenge in Africa. However, we know that biotechnology for livestock conservation and development offer potential solutions. There are many African instruments that are available to support the needed transformation. These instruments are embedded in the Agenda 2063. Now, some of you have never heard of Agenda 2063. Please Google it, you'll find out. But we are looking at the Africa we want. And we are looking to accelerate Africa's transition to an innovation-led, knowledge-based economy. And in the CAADP strategy and action plan of 2026 to 2035, looking at building resilient agri-food systems in Africa, we, as partners with the Center for Tropical Livestock Genetics and Health, and other institutions in developing countries and from the more developed economies, are looking at how livestock technologies can be used to achieve or catalyze the change we desire. So this side event is to help us begin those conversations and to look for solutions. So our objective at the end of this is to provide some roadmap or some simple factual ways of looking at how can we use biotechnologies to transform tropical livestock and also catalyze discussion among diverse stakeholders and actors within the livestock space. We also want to create awareness globally. One, that within Africa we do have solutions and we do have people who can implement those solutions. And two, within Africa we can create the right environment to harness the benefits that we have within our continent. So, with those few remarks, I'd like to introduce some speakers who are going to guide us in the different topics. Now, we're going to have some presentations. I think we have 4 short presentations. Short. I remind the presenters you have about 10 minutes to give a presentation. Following each presentation, we are going to have one person giving reflections on what the presenter has given. And then we move on to the next. Following the presentations, we are going to have a brief panel discussion. And again, the panel discussion will be led by leaders from different institutions within the continent. And responding to the same questions, we will have colleagues and partners who are practitioners in the field of livestock improvement. So we hope that we'll manage the conversations and discussions within the 1.5 hours that we've been given. So first on the podium, I'd like to request or welcome Professor Tom Burdon. Right? He is the group leader and a senior research fellow at the Roslin Institute. And he is going to tell us a little bit about what is going on in the laboratories since 2022. Is that correct?
Maybe a bit longer.
A little bit longer. Welcome, Tom.
Okay, thank you very much for the invitation to give this talk. There's a delay, a slight delay apparently on this. So in this talk, I'm going to talk about the work that we've been doing in the lab to use stem cells to study livestock biology, but also I'm going to give you two examples of how colleagues of mine at Roslin have been involved in surrogate technology for conservation and dissemination of livestock genetics. So, this is going to take, oh, there we go. Now I've gone too many. So, basically, the work in my lab, the ambition of my lab is to essentially generate livestock in a dish, and that is using stem cells to generate differentiated cells that we can then model livestock biology. And the purpose of this, the aim of this work, is to identify the genetics that underlie healthy and disease states in livestock. The workhorse in our experiments in the lab is, that's no pun intended, is the pluripotent stem cell. So this is a stem cell that's derived from the early embryo, pre-implantation embryo in mammals. And this cell, these founder cells, generate all the differentiated cells in the fetus normally. But when these cells are explanted into culture, they can be maintained and established as cell lines. And these cell lines exhibit two important characteristics. They divide unendingly. They're essentially immortal in culture without transformation. But under the right kind of circumstances, they can be differentiated into all the fetal tissues in vitro or in vivo. And so essentially you have a very nice system here where essentially you can generate a whole variety of different differentiated cell types in culture in the absence of the animals. However, in this particular case, we rely on the embryos to generate the cell lines. So in 2006, Takahashi and Yamanaka, two Japanese scientists, discovered a new way of generating these stem cells. And what they discovered and reported was that you could express in somatic cells, such as these skin fibroblasts, a cocktail of transcription factors from stem cells and convert them directly into these immortalized pluripotent cells. And they named them induced pluripotent stem cells. And essentially these cell lines are the same as you get from the embryo. They grow unendingly, and limitless production of those cells in vitro, and differentiate into all fetal cell types. So we've been using these cells to generate stem cell lines for a variety of animals. We've made them from a whole variety of very closely related animals, the domestic pig and its wild equivalents. We've also extended this to generate cell lines, how annoying that is, from wild animals as well. In this case, warthog, red river hog, and a babirusa. And the reason why we've done this is because they show different sensitivities to a really important pig pathogen. That is African swine fever virus. So domestic pigs, wild boar, and babirusas all suffer from a hemorrhagic fever which kills them within about 7 to 10 days when they get infected. But the warthog and the red river hog, although they get infected by virus, they do not die from this disease. They basically survive perfectly and don't develop a fever at all. So we're interested in what are the genetics that allow some animals to be susceptible and some to be resistant to ASF. Oh man, got to press the right buttons. Using up valuable time. So this is now a video of cells that we've derived from these stem cells, which are the targets for the virus. So this virus basically lives in the macrophages of the animals, and we've generated in vitro the macrophages here, and this video just shows how phagocytic they are. They're taking up little beads that we've added, which fluoresce when the cells take them up and into their acidic vacuoles, phagosomes. These cells are morphologically, functionally, and genetic, I would say, expression-wise markers similar to the macrophages you might isolate from animals, and they also interact normal ways with existing macrophage pathogens. And of course, the important one is ASF. So in this panel, what I show you here is macrophages that we've generated from all the various stem cells that we generate from the different animals. We've infected them, and then we stain them 24 hours later for proteins that the viruses are producing, both early in the green and late in red. And what this demonstrates that in all of these cell types, basically the virus infects normally and proliferates in its normal lifespan. And so what we're interested in is, what are the differences between the African wild pigs, how they handle the virus, and how they respond in signaling to the immune system in terms of generating a fever or not. So the steps we need to do to understand the genetic basis of this is, of course, gene editing. And in the lab, we basically deploy two forms of gene editing, or genetic manipulation. We do targeted gene alterations for specific genes, and this is just an example here. What we've done is we've knocked in, just for demonstration purposes, a GFP, a fluorescent marker, into a housekeeping gene. It's expressed in the stem cells there, in iPS cells, but also the mutation, of course, is transmitted to the differentiated progeny, and you can see nice green macrophages there carrying the gene alteration. We also want to do screens to identify new regulators of the virus. I'm too heavy-handed. Sorry about this. I hope I get some more time back. We also deploy lentivirus-based screens. And then in the panels at the bottom there, what you can see is that the lentivirus vectors infect our macrophages very efficiently, with over 95% efficiency, and we can basically introduce into these cells either knockouts or overexpression of cDNAs. In this case, in the right-hand graph, we've overexpressed antiviral genes which suppress ASF in our transfected macrophages and transduced macrophages. So basically, our plan is to use the ES cells and the derived macrophages combined with gene editing to understand the genetic basis of resilience and susceptibility to ASF. So now I'm going to switch to the two examples of surrogate technology that have been developed in conjunction with my colleagues at Roslin. This one here comes from Professor Mike McGrew's lab, which is world famous now. And so what Mike has done is basically made use of the fact that the stem cells for the germline, the primordial germ cells, migrate in the early embryo through the bloodstream. And so what Mike can do is sample the blood, take those samples, then grow those cells in culture. He's devised very specific and very nice, efficient conditions for expanding those cells. You can then bank the cells, you can freeze them down, and then also you can genetically modify them. And the key thing is that you can then reintroduce them into the bloodstream of a host embryo, and they will then repopulate the germline of that host. And the clever part of many clever things that Mike has done is he's genetically engineered a host which lacks its own endogenous germ cells. So basically, engraftment is really efficient, and what that means is that you get host chickens when the eggs hatch, where all the somatic cells are from the host, but the germline almost comes exclusively from the cells that you've had in culture. And what this allows you to do is essentially disseminate that genetics very widely in many ways. You can basically use it for conservation of local breeds that have been banked, for example. You can introduce gene editors to make disease-resistant chickens, or you can produce phenotypes for farmers, preferred phenotypes for farmers. In the second example of this surrogate technology, this has been developed by John Oatley in the USA in collaboration with Rosalind. And he's taken a later stage of stem cell development. Basically, he's grown spermatogonial stem cells. And what they do is they take seminiferous tubules, and they basically isolate testicular cells. They generate pure populations of spermatogonial stem cells, and then they transfer them back into a specific host. This host lacks its own endogenous spermatogonial stem cells, and this is a result of a particular gene mutation that they've introduced. And what this creates, essentially, is a host, an animal, which has basically the phenotype could be of an extremely resilient animal that's suitable in a particular environment, but it carries in its gonads, essentially, the genetics of a genetics that you might want to introduce into the breeding population. And what this allows you to do is essentially disseminate this genetics in a very similar way as an alternative to IVF. So I just want to finish off by introducing something which is a bit more fanciful. Sorry about this. It's my control of this. Obviously, it's my shaking hand that's the problem. Wish stem cells could cure that. Anyway, basically, in cattle, productivity in cattle, reproductive cycle is about 18 to 12, 24 months. And this basically provides a limitation on the kind of genetic gain that you can get in time. And what's been proposed is that by combining stem cells and germ cell technology, essentially we can increase that rate and create a cycle of in vitro breeding, essentially. So you generate stem cells, which we can do. You generate gametes, which we can't yet do. You can generate artificial embryos in vitro and then generate more stem cells. So basically you can drive the cells through this cycle through repeated rounds of meiosis and gamete production, in theory, and essentially accelerate the generation of new combinations of genetic variation. And in combination with genetic selection, you basically can accelerate genetic gain in your breeding population. Now this structure, this somewhat fantasy in cattle at the moment, has been demonstrated in mice, so it's not been done in any other species. It obviously has some very direct implications in cattle breeding, but also has important implications for conservation technology. So potentially one might be able to resurrect animals by generating the gametes in vitro and basically save them that way. So what I'd like to conclude then is I think that ES cell technology combined with germline and reproductive technologies will provide, I hope, in the future, a way of improving the resilience and the productivity of livestock in the future. Okay, that's my talk. And I just want to finish with acknowledgements, of course. So basically, these are the people that have done the work, and also I would like to acknowledge the CTLGH funders.
Thank you. Well done. Thank you very much. Thank you for keeping time. I'd like to request Dr. Victor Olori. Is Dr. Victor Olori online?
Yes, please.
Thank you. Dr. Victor Olori is a senior geneticist at Aviagen with the responsibility for the group's genetic evaluation. Right now, Victor, from what you have heard in this presentation, could you give some response or feedback or on its applicability within the African livestock systems?
Thank you, Julie. I hope you can hear me.
Yes, we are hoping to see you.
Okay, I'm on screen, online, so hopefully. I think without wasting time, what Professor Bolden has shared with us, it's the tail end, or what I would refer to as a highly technical methodologies or technologies that will help us. Okay. Am I all right to continue?
Please go ahead.
Okay. So he has shared with us just a few of the high-end technologies that are already being developed, hoping to fruit in a few years, that can help us do three very great things. One, to conserve the expansive genetic resources we have in Africa. Some of these technologies can help us modify and quickly improve the livestock we have, while also some of these technologies will be very useful in helping us to disseminate genetic gain when we get it. However, what I was asked to mostly mention or discuss today again is that concept of, how do we prepare ourselves to be able to utilize these technologies whenever they become readily available so that we can be one step ahead. And so I'll be able to go back to just my discussion. We know that Africa is awash with livestock genetic resources, and that there's a lot of species, a lot of breeds, a lot of populations, and even individuals. Within each individual, there's a plethora of genes which determine their features and performance. For decades, we have failed to harness this genetic variability to produce animals that are physiologically efficient in production and in health. Inefficiency in livestock production is no longer sustainable or acceptable, especially with climate change. So the deployment of science and technology, and some of the technologies described here today, can help us change this narrative. Now, farming is a means by producers to provide animals with a good environment in order to protect, nourish, and hence improve performance. However, improving the production environment alone cannot lead to a permanent and transferable change in performance. Such change can only be achieved through genetic improvement. It allows the accumulation of desirable genes in a sample of populations used to produce the next generation through various methods. Some of the methods is what Professor Borden has described, but genetic improvement of individuals can also be achieved through selective breeding. This means identifying individuals with superior genotypes and breeding only within these individuals. When this is repeated over many generations, we are able to obtain animals that perform above the population average, both in performance and in health. Success and rate of gain is dependent on the genetic variability, the intensity of selection, and the accuracy of selection. Now, genetic variation and selection intensity will be high if we have a large population to select from. Accuracy of selection depends on how well we can rank these selection candidates on the basis of their genetic merit. Pooling resources across countries in Africa will be beneficial for both of these things. Selecting individuals with superior genotypes also require us to be able to rank all animals, all available candidates on the basis of their genetic superiority. But genetic superiority is not always obvious to the naked eyes. It requires genetic evaluation, utilizing performance and genomic data, and some of the things that were described earlier on today. Key requirements for a genetic evaluation are the phenotype, the genotype, and the pedigree. In addition to these, selection requires a definition of a breeding goal and identification of the traits relevant to that goal. This requires leadership, human capacity, and some basic infrastructure. A structured breed program operated by knowledgeable individuals of various capacity is capital intensive. It requires investment, dedication, and collaboration. The lack of leadership, poor human capacity, ignorance, and poor investment is the bane of animal breeding in Africa. However, pooling resources and capital across countries will allow us to overcome these difficulties and evolve a continent-wide breeding program for livestock improvement. The African Animal Breeding Network has risen up to provide this leadership to solve the human capacity problem, facilitate genetic evaluation, foster advocacy, and business incubation. This will make it possible for the best genetics in any one country to be accessible to farmers in the other country, hence allowing selection and breeding across the vast genetic resources that abound across state lines. Our vision is to bring about sustainable genetic improvement in Africa by driving development and dissemination of genetically improved seed stock. We cannot do this alone. It requires the collaboration between all stakeholders. And I think that's what what is on the screen that you can see is saying.
Thank you.
There are challenges which also provide business opportunities that need to be discussed. Hopefully we can do that. How do we get countries to participate and collaborate? How do we source capital for farm unit development? How do we standardize data collection across countries? Who will fund the central unit for data management and genetic evaluation? How do we set up the infrastructure for distribution of genetic gain? What some of the things that Professor Borden has described today, when they become fruitful, how do we get this to all the farms across Africa quickly?
Okay.
So, above all, how do we disseminate knowledge of the principles of selective breeding and develop the human capacity to see this through? Now is the time for us to discuss and chart a way forward. Thank you.
Thank you very much. Thank you very much. I think very, very deep questions. Let's appreciate Dr. Victor. Thank you, Victor.
Thank you.
Yeah, very many questions. As we said, we gather in the evening to deliberate and to review. What can we do? We change tack. I'll ask our second presenter, Professor Raphael Murode, to come and share his thoughts. Now, again, we are going to a different topic because there are many issues. Raphael is a professor in quantitative genetics. He's based both at ILRI and at the Scotland Rural College. Many of us have used his book as a guiding principle in our learning genetics. So Raphael, you are a tutor to all Africans, and not only Africans, to all geneticists across the world. And today we want you to tell us about climate-smart livestock development in Africa. Welcome, Raphael.
Yes, you will have noticed a bit of change in the topic. So I'm just going to spend the next 10 minutes talking about moving from multi-country genetic evaluation to exploiting genetics to mitigate environmental impacts. So just first of all, what are the characteristics of a climate-smart livestock system? The components are improvement in productivity and efficiency, improvement in fitness, improvement in welfare traits with little or no negative impact on the environment. In summary, in brief, those are the characteristics of a climate-smart livestock system. So the question is, why is a climate-smart livestock system vital to Africa? It is because of the vital role that livestock actually plays. In sub-Saharan Africa, for instance, the livestock sector contributes 30 to 80% to agricultural GDP. Around 70%, over 150 million people actually rely on livestock to sustain their livelihood. I just gave a few examples here. Kenya, for instance, the dairy sector employs 1.8 million small householders. Direct employment for about 750,000 people with additional support for 500 individuals. However, because of the anticipated increase in population of Africa, the need for animal source resources is expected to double. The second reason why the climate-smart livestock system is essential because climate change is upon us. And it's already having impact in terms of feed resources, the emergence of new diseases. So although right now the contribution of the sub-Saharan Africa in terms of methane emissions are around 10%, but given the significant changes we anticipate in terms of growth, given the anticipated change in terms of the demand for animal source food feed, we think that this could actually triple if we take no action. And more importantly, we have limited data and the capacity right now in Africa to actually measure methane emission. So given these factors, a climate-smart livestock is inevitably a priority. So why the application of genomics and multi-country genetic evaluations? Because genetic improvements are permanent and they are cumulative. We need genomic approaches so we can understand the genetic basis that drives these traits, and therefore we can actually put in place targeted interventions. A multi-country approach is necessary because data is limited. We need to put data together. We need to increase accuracy and to be able to select superior bulls across the whole continent. And the benefit of that have been demonstrated before from several studies, and I'm not really going to go into the details of that because of time. Just to give an example that increase in productivity is already happening in Africa. So these are some of the results from the Africa Dairy Genetic Game operating in these countries. We use a digital tool to collect data. We use genomic predictions. We have a restricted index to improve productivity and restrict body weight gain. It means that we are actually selecting more efficient cows without increasing the amount of feed they actually consume. And this is one of the graphs that show the change in the trend for making it in Tanzania. When we started, it was around 6 point something. Now we can see an increase of over 10 liters per day. And the consequence of that is that as we increase efficiency, the amount of methane or carbon equivalent per unit product actually decreases. So improvement in productivity efficiency indirectly actually reduce the carbon footprint. Now I'm going to talk about genetics to directly select for improving or reducing environmental impact. Such a research must be multifaceted, addressing issues of mitigation, adaptive capacity of animals, and must ensure increased productivity and efficiency. And so we have at IRRI, we have this project we call the EnviroCal project, which is using, it's a bit slow to respond, using a very multifaceted approach, looking at direct selection for methane, improving efficiency, and increasing the general potential for resilience. So how are we doing that? We're measuring methane. Right now, we have methane records in over just very close to 1,000 cows in over 80 dairy farms in Ethiopia and Tanzania. We're using the LMD. It is portable, and that is why we have used it. But the beauty of it is it can give you measurement over the life cycle of a cow. Indirectly, at the same time, we collect a mixed sample, and we can get the mid-infrared information. So we want to be able to indirectly predict methane without having to use the LMD. And we are using body weight as a proxy for feed efficiency. We're using THI to increase heat tolerance. And overall, at the end of the project, to develop an index to improve efficiency with less impact on the environment. So what are the results we have obtained so far? So from the study, we see that we have heritability of 0.2. That is very good. It means that, for instance, if you want to have a bull with 85% reliability, we need just 100 daughters. Then if we indirectly predict methane, just using 5%, 40%, we have an accuracy of 0.43. When I mention this figure, people say, oh, that is very low. But you know what it means? It means that I only need 300 daughters with milk records to be able to get a reliability of 85%. And I think that is very good. Now, there is a coordinated approach. So IRRI have just won another project from the Methane Hub. And we are going to work in East Africa, in South Africa, and West Africa. We are going to measure methane in over close to 3,000 animals, dairy, and 180 beef animals. They'll be on-farm and off-station. We'll do microbiome profiling. We design a genetic model to actually analyze the data across the 3 regions of Africa. And that is where the multi-trade country evaluation comes in. And we have opportunity to exchange data with the global north and eventually develop an index. And just to go back to actually reflect something that Tom already said, we can actually increase our effectiveness if we combine this with reproductive technologies. So there are 2 factors that drive genetic improvement: the accuracy of genomic prediction and the intensity of selection. If we use assisted reproductive technology, we can reduce the generation interval. And so this is something I think that was published very recently in the agricultural magazines, a work done at SRC called the Cool Cow Project. I like the name of the project. They took eggs from a cow and they fertilized that with a genetically superior bull selected for methane reduction. They carried out 80 and so on and so forth. And they were able to reduce generation interval to just 8 months. And of course, Tom is giving us a much shorter time, but his own is just theoretical, but this is already done in practice. I'm going to now conclude. Climate-smart livestock development are inevitable as we look to the EU agenda for 2063. If livestock is to continue to play the major role it's already playing in sub-Saharan Africa, a multifaceted approach applied genomic method is essential, with productivity and efficiency optimized while reducing adverse environmental impact. And for us to do that, we need a multi-country genomic prediction system. Important to get the maximum benefit for such difficult to measure traits such as methane. Data exchange is a major component of all projects by the Methane Hub. If you are not willing to sign for data exchange, you're not going to get $1 from the Methane Hub. It means there's the need to actually realize that within Africa, we need to exchange data, very critical, and multi-country evaluations important. We need national evaluation system to deliver the last mile in terms of that superior genetic that will come out of our system. And for us to get the maximum benefit, we need to add to this the use of reproductive technologies, ET, and so on and so forth, so that we can actually improve productivity and reduce environmental impact quite quickly. And just to end up acknowledging the contribution of my workers, Keyo, Gabriel, Julie, and Shirley, and all other workers. Thank you very much.
Thank you, Raphael. He has the height advantage over me. Thank you also for keeping time. Now, in response to what Raphael has shared, I'm going to request Professor Sami Agri. Sami, are you online?
Yes, I am.
Excellent. Professor Sami Agri is a professor of population and quantitative genetics. He's a faculty member at the Department of Poultry Science and a member of the Institute of Bioinformatics at the University of Georgia in USA. Now, Sami, I know you were given questions to answer, but before you answer your question, could you give a brief response or review on what you have heard from Raphael, and then go on to answer your question in 5 minutes? Thank you.
Thank you, Julie. What I would say with respect to what Raphael said, the bottom line is we have to work together as a continent, pulling data and resources together in order to succeed. Going individually may not be the best approach because the data that he has analyzed across countries have shown significant success, and that would never have been seen or shown if each country had to go alone. So that would be my overall general response to what Dr. Mrode said. And now I will shift to what kind of vision or models should we have for our curriculum? And I would say I'll probably step back before I step forward. Our training classically has been a data collection process, which in the West has been done by breed associations, companies, cooperations, and that is the basis upon which most of us studied. And all this is done with one goal, financial gain. So animals are kept for financial gain, so all the models go around that. And we always have extremely large data set that are connected. When I say connected, it means there are some genetic relationships within the data set that is collected. And this is supported by genetic models, statistical tools, computations, and in the last 20 years, as we progress, we've added genomics, and most recently, we have AI coming into the forefront. Now, let's see whether this type of curriculum is still needed for the African continent. In Africa, when we talk about livestock, we have financial goal as an objective, but we have other goals as well. For the purpose of this crowd, I will just concentrate on genetic improvement. Now, let's talk about data. From Dr. Mrode's speech, make mention of smallholder farmers in Kenya, I think 1.8 million. So in Africa, we're going to get small data sets, as opposed to the West, where the data sets are large. And also, the data is going to be sparse. And the data is going to have connectivity issues. So these are some principal things, practical things that are on the continent that we have to deal with. And how do we get around this? Which means we need some machine learning techniques for small data set. So students, the curriculum has to include large portions on how to deal with small data sets because they are different topics on its own and it has to be well covered. And how to do transfer learning, simulating data and validation. Because you're not going to have the 10 million records that you need, 5 million records that you need. And sometimes you have to model traits, make adjustments in the data. I'll give you an example. You want to go collect, let's say, body weight of chicken. Somebody may collect it at 6 weeks. Others may collect it at 4 weeks. Others may collect it 12 weeks. Milk the same. You need some training that would allow you to model this trait to standardize them for breeding or genetic improvement. So data handling and adjustment becomes also crucial. In genetic improvement, bad data also means that you're going to get bad output. So it's important that you get the data part correct. And again, you also need statistical tools that can handle these structures, and you need computational skills. What I would say is that genetic improvement is a quantitative science. So if you don't treat it as quantitative science, then the training will be highly inadequate. So therefore, we have to understand that going forward, we have to treat it like math, statistics, programming. It is not all about, I would say, decision. There's a large proportion of it that is really quantitative. So what I would say is that I would like to see a curriculum being kind of written or thought around 5 different categories, which I'll call the 5-star training.
Yep.
That the first one is data science.
Yep.
So all animal breeding students should go well versed in data science, 2 statistics, 3 computation, 4 quantitative genetics,
Yep.
and 5 selection and breeding strategies, which are probably the decision-making process, supported by genomics and AI in recent times. So that is what I would like to see where the curriculum goes, and I think I would stop here.
Thank you, thank you. Thank you, Prof. Wow, we have heard from 4 distinguished leaders. From the audience, is there any brief or very quick question or observation that somebody might want to raise just so that we can break the hearing? Was a hand there? Yes, go ahead.
Yes. My name is Chinyere Ikini-Jivenu. I work as a statistical geneticist with the Livestock Genetics Group at ILRI. And just to re-echo what Dr. Sami Agri said with the curriculum, is what we were discussing this morning, and I think it is past time that we revised our curriculum on the continent to support the kind of work that we need to do going forward. So just to say that this is something that should start happening right now, is that we revised our curriculum, starting with the trainers, so the faculty members, and then they would transfer it to the students.
Thank you very much. Thank you very much. Any other observation? Yes?
Yeah, thanks. I would like to pick up and re-emphasize one sub-bullet point from Raphael's presentation. He made the point that a major constraint is distribution of genetics. And I think it's really important to realize that even if we had, I don't know, 100 breeds of absolutely perfect animals, perfectly adapted to 100 different environments, the constraint would be getting them out there. And if I was planning research, I wouldn't be plugging broken liquid nitrogen systems. I'd be looking at novel methods of delivering those genetics. And that's actually where the surrogate sire technology has an additional advantage over its speed of reproduction.
Thank you. Okay, one more, and then we go back to the presentations. Yes, sir.
Thank you. My name is Oluwafemi from University of California, Davis. I just want to make this contribution about the Global North and Global South as a student who have experienced the Nigerian system. I'm originally from Nigeria and also US. I think there's a lot of gap between Africa and the developed world. If we really want to bridge this development increase we want in our genetic reservoir, I think it's high time we started, maybe the government started investing judiciously in helping even labs to actually have what we need. Taking, for example, many big universities in Africa do not even have a good lab, one good lab. And when you get to US, you have just one professor having like 3 good labs and they are doing things. So definitely there will be a lot of gap. And that is why you see many of our professors who are Africans, maybe they are not willing to come back home. And we need to encourage them to come. Thank you.
Thank you, thank you. I see you're from the next generation. I'm very happy you're going to be our professor with the new lab in Nigeria. So welcome back. We move on again to hear again from our colleagues from the north. And I'd like to welcome Professor Tad. Now, I will keep quiet on the second name because he told me and I've forgotten. Sonstegard. Yeah, I'm a good learner. Right, he's an internationally recognized leader in livestock genetics with more than 20 years of experience in the field. He's made contributions to the sequencing of livestock genomes, development of industry-standard genetic diagnostic platforms, and elucidation of the genetic basis for traits impacting food animal health, production, and well-being. Welcome, sir. Let me adjust the height.
Thank you for the introduction, and thank you very much for the invitation to participate in this meeting. What I'm going to talk about is really to inform you what we have already learned about the past more than 12 years that we've been trying to take this technology of gene editing and turn it into a breeding tool for animal improvement. And I hope that this information will help your discussion about the Africa we want relative to cattle. And I didn't just come all this way for giving a 10-minute talk. I think it's really important that our company is here representing, having face-to-face discussions with you, in part because, let's see if this thing moves. Go ahead, hit it. Okay. If you look at our mission statement, really what Exceligent is still about, even though we're now been acquired by Transova, and we're a part of the Eurus Alliance, a big genetic company that's global, is we still have the ability to share and partner our technology to help breed better cows. I have to show this slide because maybe not all of you know who we are. We're a very small R&D company located in Minnesota, from a group of professors at the University of Minnesota that founded a platform to gene edit animal cells. And so we run very lean, only 18 employees, and we think we've been a global leader, at least in gene editing livestock. As far as animal goes, it looks like Colossus took the lead in that, but they outnumber us 50 scientists to 1. So, if you want to develop a breeding program that uses gene editing technology, which is really the next logical step as far as breeding tool goes, being able to write back and recode DNA to put in the phenotypes you want in your animals, you really need to have all of these technologies available, and that really involves a breeding plan. And that breeding plan needs to be based on the voice of the customer, and Professor Kemp made a very important point: you need to be able to distribute it. Doesn't necessarily always have to be through AI technology, which is really something that came about in the 50s. It could be a different type of breeding plan where you're actually creating animals that are off the shelf that can go out and do that breeding. So supporting our claims to expertise in this field, we have a lot of traits that we've worked on. We've worked on many different cell types. We've knocked genes out. We've made precise changes to things that already naturally exist but just aren't in our current breeding populations. And so this timeline demonstrates our work over the more than past 10 years. We currently have 8 bovine traits that are in our commercialization pipeline. 3 of them have already been allowed in 4 different countries, and we already have semen cells in the US, Brazil, and Canada. We do have dossiers that we have prepared and have submitted to Kenya, and we've also prepared one and submitted to Nigeria, and that's for the heat-tolerant Holsteins. Next slide, please. So what have we learned from this past decade? We can take elite performance genetics that aren't meant to be in the subtropics or tropic production environments, in part because those cows aren't comfortable, and we can adapt them to those environments, especially for heat tolerance. As far as taking indigenous genetics and trying to improve production, I think that's still a ways off because there's still many, many genes involved in a lot of those quantitative traits, and you have to be able to do really high throughput multiplex editing in order to do that. I'm not saying it's not possible, but the cell technologies that we have are going to be critical for being able to do that multiplex editing. Next. So really, where we're focused with this technology is the promise to take the best genetics, match it to that environment, and also be able to match any management issues that may be in that environment. Next, please. So I'm going to give a commercial example of that. If we look at slick Holsteins, so these would be the heat-tolerant Holsteins that would be more optimal for tropical production versus wild-type Holsteins. And we look at genetic trend. Right now, there's only 16 slick Holstein sires that are actively marketed semen-wise, and all of these are heterozygous, so only 50% of their daughters are going to have that trait of being able to withstand heat better. The comparisons have been done, though, with studies by Livestock Improvement Corp, is that those slick daughters maintain a 92% conception rate versus the non-slick daughters, which are very much reduced. So that's a trait that puts revenue into the farmer's pocket. They also produce more milk as far as pounds per day. If it's even 5 pounds more milk per day, that is a game changer. So there's data from the University of Florida, and we suspect there's going to be a lot more data coming out here as studies on conventionally slick-bred Holsteins continue. The problem is, of course, if there's only 16 bulls, farmers don't have a lot of choices before they run into an inbreeding problem if they were to continue to use slick. Next. And this is just data from Puerto Rico, again, emphasizing those differences in the fertility and the milk production because of that buffering for the heat tolerance. Next. So if we look at how these bulls compare to genetic trend, there's really only 7 of these bulls now being actively marketed, and not all of the semen companies sell them. There's 0 proven daughters, 0 daughter-proven sires, excuse me. So that means we don't have progeny tests. And nobody does genomic evaluations based upon whether or not you have slick or not. It's still the traditional genomic evaluations based on production of whatever genotype you are. So slick bulls really only make up 0.2% of those bulls that we sell semen on. So why is that important? If you look at the averages in the genomics, which is something that policies are concerned about as far as importing genetics, the genetic trend of these slick bulls is 6 years behind. But that difference in having that trait for slick has a major effect on production. So those genomics really aren't comparable to what's out there for the traditional genomic evaluation. Now, if we're able to do a homozygous slick edit, we can change, in a top 100 bull, we can change that genetic lag to be only 1 year, because we can take a cell line from a calf and produce that, a clone of that calf that is homozygous for slick, and now we're only 1 year behind. So that's really what we're working on right now. Next. We do have homozygous slick bulls that are already available to be marketed. In part, that was through our Gates-funded project and also projects we did internally. And I would point out that if you look at the traditional indexes calculated by the genomics, I've highlighted 3 here where they're negative. If you were to evaluate importation based upon these are not traits that bring revenue into the farmer's pocket. It's the slick is what they want, and those daughters from that slick. So these bulls are still very strong and competitive amongst slick bulls. And so this is something that needs to continue to be evaluated until we get into those bulls that are within the top 100. Next, please. Next. So with heat tolerance, especially in the Holstein breed, we can adapt those genetics to help those high genomic bulls reach their full genomic potential in a tropical setting. But just the heat tolerance itself, even though it's a better animal well-being and the animal's more comfortable, there's still diseases that it's susceptible to. Next. So we really wanted to focus on improved health, especially for those types of diseases where vaccines have been ineffective. Next. So we thought the best way of doing that, of course, is moving into stacking traits. So doing more than one edit at a time based upon naturally occurring alleles. Some of those alleles, which we found segregating in West African breeds, some which are very common in Zebu breeds. And then we've also been working on other non-GMO solutions where we know what the receptor for the virus is, and we can just snip out pieces of that virus or change amino acids, and then that virus is not able to infect those animals anymore, which would be our bovine viral diarrhea virus project. Next. Next. Okay. So that brings me to the Thumani breeding project. So that was just a picture of one of the animals we created. So we developed a breeding plan, and we're going to de-risk through an investment from the Gates Foundation the ability to multiplex and also use cells that have never been used before as far as editing and going through a cloning process to make an animal. So we started off by picking 2 different breeds, one that's adapted and one that's not, and using a genetic base that is friendly for gender equity traits. And when you cross these 2, of course, you could end up with something very similar to what they have in Brazil with an F1 Girolando, so we selected Holstein and Gir. And then, of course, if the Holstein is slick, you have a difference in case use where you can actually go up to 3/4 Holstein, so we're not dead-ended at just making an F1. We wanted to focus on local disease traits and heat tolerance. The key here is we needed durable cells in culture in order to do this multiplex editing. You can't just throw 4 different CRISPR guide RNAs in and target them. Okay, next slide, please. So very quickly, our process is not cloning animals, it's cloning IVF embryos from high-value genomic matings. Next. Go ahead and click through 6 or 7 times. We can take and create an embryonic stem cell line. We can do genomics on that line to pick the best line for doing the editing. We can do the editing. We do nuclear transfer from those cells. Next. And we've created multiple bulls from that. So what have we done? We've broken the barrier for durability of cells in culture because most cloned cells don't last very long. And we're able to do that multiplex editing. Next, please. Next. So these are 5 of the sires we created. All of them have produced semen for exportation from the US. Next. We also have females. We've created IVF embryos for export using the sires to create those homozygous embryos. Next. We've also made GIR animals. Next. Those have 4 edits. So they have a Holstein allele and a trypanosomiasis allele. And these GIAR actually shown on this screen are actually the highest for milk production and early precocity in Brazil. Next. So commercial deployment is challenging, but it's still possible. So what are the impediments? It's really, in my opinion now, politics that influences methodological policies about the risk of this technology, and we're still dealing with it in the US in trying to get FDA to realize this is really a low-risk technology as far as food safety and environmental safety goes. Next. So I threw up this last slide as a discussion point. You know, we've spent invested over $100 million developing these core technologies and this trait discovery, intellectual property, taking what we learned from human biomedical and from our academic collaborators to develop a commercialization pipeline for all of these different technologies just to make one animal. I think when you think about where you want to enter in this, do you want to build this whole system, or do you want to partner with us by taking this top part of the commercial plan and your federated databases that you're talking about and really think about what animals do you want to edit and what traits do you want to bring into those that are things that the farmers need. Next. Okay, this is just acknowledging all the people that have worked with us on all of these projects. Appreciate your attention.
Thank you. Thank you very much. Thank you. Thank you. Thank you. Thank you. Yes, he traveled a long distance. You're forgiven. Thank you very much. Now, we have Professor Alexander Kahi. Are you online, sir?
Yes.
Excellent. Professor Alexander Kahi is homegrown, homebred professor in animal genetics and breeding, and he also works from home, which is excellent, right? He's a professor at Egerton University, based in Joro, Nakuru, Kenya. He's also the director of the Center of Excellence for Livestock Innovation and Breed and Business for the continent. So Professor Kahi, you're very welcome. And before you respond to the question you are asked, could you give some remarks and comments on what you have just heard and on its applicability within the continent of Africa? Thank you.
Okay, thank you. Thank you very much, Julie. And let me take this opportunity to thank the organizers of this conference for inviting me to be part of it. I've listened to that presentation on genetic engineering and assisted reproductive technologies. And as we are all aware, I think these are very important technologies that we in Africa, if we adopt them and use them, then that will go a long way in improving our livestock within the African setting. I really liked the way it was presented, and also liked the fact that this is a presentation with a sort of a commercial angle, and this is where we should be going as Africa. And it is my feeling that with the ABNet and within the advocacy, awareness, and business development pillar, it is my hope that we can really work very hard towards ensuring that we end up with a lot of businesses that use some of these very important technologies for the betterment of the African livestock. But that is a very good presentation, and it is a very good form of encouragement. And what I would like to say is that I think there'll be need for us to partner with such companies so that we can also use these technologies as African scientists. So that is what I can say about the presentation, but it is an eye-opener, and let us as ABNet try and think very hard on how we can ensure that we have businesses that can support animal breeding in Africa. So that said, My presentation is on just one of the pillars of ABNet. I know the previous presenters talked about the other, I think the other two pillars. And with me, I'll be talking about one of the very important pillar, and that is advocacy, awareness, and business development. When you look at the slides, I think the slides talk about what ABNet is. I think I will not really talk about that because that has already been mentioned. And then the slide also talks about an overview of the livestock sector, the importance of livestock in Africa economy, and also talks about some of the current challenges that are facing the livestock sector. But I will not talk about those because of time. What I'd like to concentrate on is what is this that we really intend to do or we are doing as ABNet within the advocacy, awareness, and business development. I think you are all aware that ABNet was launched in June 2021. And from that time, it has promoted livestock genetic improvement programs among key actors in the African livestock sector. And what ABNet does is simply drives advocacy through policy engagement, representation, as well as through partnership initiative to strengthen the livestock investment ecosystem. And this network, as we are all aware, it drives awareness through capacity building and information dissemination. And as we speak, we've already had one training here in Kenya where we invited, I think it was over 40 people who are trained in different topics in animal breeding. And when you look at this particular pillar, business development is very important. And ABNet promotes business development through market linkages, all this innovation, adoption, and also through promotion of investments within the livestock value chain. So this particular pillar aligns very well with the COILIB's expertise in sustaining Pan-African livestock transformation. So when you look at COILIB, COILIB stands for Center of Excellence for Livestock Innovation and Business. But when we started this particular center, it was simply a livestock center, but people complained because we were incubating only businesses within the livestock sector. And because of the fact that this was the first business incubation center around, we had now to change, not really to change focus, but simply to ensure that we also incubate businesses within the other sectors. And because of that, what we did, we simply added a comma between livestock and innovation. So currently, our focus is on livestock, but we also do innovation and business development in the other sectors. And we have three pillars. The first one is on incubation, leadership and market development. And then there is another one on information, technical assistance and consulting. And then we do research and commercialization. And then at the end of it all, we have a very big and very important pillar on communication, media, and advocacy. At COILIB, of course, we have a TV and a radio station. So when you look at what we've done, because of time, I will not really talk about everything, but we've been in existence for the last 15 years, and we have reached across 49 African countries, and we've reached over 18,000 farmers. I don't know, can you go back? I'm saying you're playing the video. I think this is not the time for the video. Yeah, I think we are still, just back. Okay, there we are. Okay, so on advocacy, the next slide, please. On advocacy, of course, We have a number of ongoing initiatives and campaigns that we lead. And these are some of the campaigns that, of course, ABNet can also benefit from. We do all these youth agripreneurship programs that are financed by a number of organizations.
Okay.
And we go to the extent of influencing policy at national and international level for sustainable agribusiness growth. And as we speak, we've been part of a number of initiatives that are geared towards developing a number of innovation spaces at higher education institutions in Kenya and in some other countries in Africa.
Yes.
So we have very strong awareness programs that are aimed at raising awareness about livestock issues and opportunities. And I will not really go into all those programs, but the thing that I'd like us to note is that while doing all these initiatives, we don't forget the stakeholders, because we all know that stakeholders are very important in whatever thing we do.
Okay.
And to that end, of course, we engage stakeholders to promote livestock transformation. So when you look at the vision for Pan-African livestock transformation, I attended the session today in the morning, and the vision that we have, I think, is the same vision that was presented today in the morning, where we are talking about looking at scenarios where we'll have Pan-African joint centers for livestock innovation and business in Africa. And we really want to be part of and really contribute to the animal seed biotechnology work package of the African Union. And I'm happy that we are doing that currently. And we are really thinking of genetic improvement through Pan-African breeding programs. And here we are looking at maybe coming up with a sort of an interbull for Africa.
Yep.
And then to add on that, there is also building the capacities of the different centers of excellence that are specifically associated with the gene banks for the African animal genetic resources. And to achieve this, I think that is a vision. I think there'll be a need to focus on a number of areas, of course, taking into account sustainability issues, taking into account technology adoption, and also taking into account maybe whatever policy reform that is needed in the respective countries. So we are looking at genetic improvement through a Pan-African breeding program to increase yields and adaptability. We're also looking at creating and leveraging multi-stakeholder networks, all these living labs, and also innovation platforms, as well as building platforms for knowledge sharing and financing. So what we really need from all of us is that ABNet is a new baby, of course, maybe around 3 years old. So we'd like to encourage all members to get involved. We have a website. So if you're an animal breeder, it will be good for you to register in the website so that we can do all this together. And when I look at the plans, just finally, when I look at our plans that we really need to do next, we really need to scale all these business cases and models from science-based research, and also there is need to support farmer-based innovative approaches to the livestock improvement. And then as ABNet, we'll be supporting research development, and I talked about commercialization through targeted grants and linkages for sustainability. I think let me stop there because of time, but thank you for giving me this opportunity.
Thank you very much. Thank you very much, Alex. Thank you very much. And for this particular session, our last presenter is Dr. Billy Ratemo. Is he online or he's?
Yes, I'm online.
Thank you very much. Now, he is the program officer for genome editing at the AU Development Agency, NEPAD, where he is responsible for generating technical content on genome editing to support research and development systems, organizing engagement sessions for experts, developing and adapting curricula, and fostering cross-institutional and public and private engagements in pilot countries. Please, sir, you have exactly 10 minutes. At 8 minutes, I will stop and I'll put you on a warning mute so that then we can stop at 10 minutes. Thank you very much. You're welcome.
Thank you. Thank you, Julie, for those warm welcoming remarks, and good evening once again to panelists and experts and all attendees for this session. So my task tonight, actually, it's very simple. I'm actually going to run all of us through what Agenda 2063 is, what are the aspirations, how do they link with the livestock biotechnologies, and I'll talk a little bit about the second 10-year implementation plan for Agenda 2063. What is the significance of this sector? What is the impact of the biotechnologies? How does it synergize with the AU initiatives? What projects do we have across the continent, obviously being done by Africans? And then how can we sustain impact? So quickly then, when we talk about Agenda 2063, I'm sure most of us have heard about this, but for those who have never heard about this, is basically a blueprint or a master plan and a framework that was put together by our heads of state, basically to transform Africa by the year 2063. So the main goal is to how do we integrate all member states of Africa? How do we make it prosperous, peaceful, and obviously driven by its own citizens? So Agenda 2063 was conceived actually in 2013. It's broken down into implementation plans, which are 5. So the first 10-year implementation plan was running from 2014 to 2023. Then the second 10-year implementation plan from 2024 up to 2033. So right now we're in the second 10-year implementation plan, and I think I'll talk about it in subsequent slides. Next. So Agenda 2063 actually has 7 aspirations. So the first one is talks about how can Africa be prosperous. The second one, how can we be integrated? It talks about good governance. It talks about, how do we become more peaceful? How can it be more? We can have, security for everyone. It also talks about Africa having strong cultural identity, and also it talks about how development needs to be people-driven, and especially by the youth and also our women. So there's the gender aspect there. And lastly, how the Agenda 2063 also, talks about how Africa can be more influential, obviously, again, driven by its own citizens. Next. Now the linkages actually from the Agenda 2063, as I said, we have 7 aspirations and 20 goals. So when you look at the livestock biotechnologies, how do they link up with the goals? You see very strong linkages on goal 1, which is improving standard of living, because when you look in the African ecosystem, livestock is part of our culture. And then under goal number 3, livestock actually contributes to improving health and also nourishment of its citizens. It also contributes to transforming economy and jobs, because in Africa you see most people are smallholder farmers. And then it also strongly contributes to realization of goal number 5, that is to modernize agriculture, the livestock biotechnology itself. And also it contributes to the issue of realizing environmental sustainability. As we know, agriculture actually is a big emitter of greenhouse gases. So livestock being part of our culture also, we see a strong connection there. It connects to the issue of cultural renaissance of the African continent. And lastly, it also contributes to the engagement, the empowerment of the youth and the children. Basically, again here, looking at a typical rural African household, they have livestock, cows, chicken here and there, which are normally used, for to cover unforeseen expenses, obviously through trade purposes. Next. Now, the second 10-year implementation plan, actually, if you look at the theory of change, which has been released recently, is actually the moonshot interventions on the left side of your screen. You can be able to see them. They've been drawn from the 7 aspirations now. These are the strategic interventions which are a priority for this second 10-year implementation plan. But how do you do this if you come up with a program? So you need to anchor them through the implementation pathways. That is the continental frameworks, which is CAADP at the moment, and also the regional frameworks, which are normally domiciled at the regional economic communities, and which are, at the moment there we have 8 regional economic communities. But then again, it's also important to link programmatic activities with the national development plans, and in this case, looking at the agriculture sector as the main anchoring area. Then when you look at the enablers for programmatic enablers here, actually, you need to look at partnerships, financing mechanisms. You need to look at how you coordinate all those programmatic activities. And then another issue you need to look at is communications and advocacy also with different stakeholders. How do you do capacity development, and also what type of technology? And in this case, we have livestock biotechnology in the picture. So all this needs to be cascaded down from the global, continental, regional, national, and also at the subnational level. So when we do this, then we expect that we'll be able to contribute to the realization of the moonshot outcomes that you see on the right side of your screen, the 7 ones that I've listed before. Next. And then the significance of the sector, fellow panelists here, I think they have clearly outlined this. The contribution of the sector, more than 35 up to, I think we've seen it up to 80, especially in the GDP of the agriculture sector. The livestock also is an important for, food and nutrition, for capital generation, source of employment, social cohesion, especially during, maybe an example that comes quickly in mind is maybe during marriage ceremony, there's an exchange of livestock amongst African societies. It can also contribute to, agro-industrialization. And a good example here is, the leather industry. And lastly, it's also as a very important source of, biodiversity. In Africa, there's a lot of, so many varieties, so many breeds of, cows, chicken and all those. Next. And then looking at the impact of these livestock biotechnologies, actually, they can make interventions in terms of reproduction in combination, of course, with the assisted reproductive technologies. They can improve on animal health, an issue like resistance to or tolerance to trypanosomiasis, and also animal nutrition, the amount of food that maybe animals can be given so that, they don't emit so much gases while they are consuming these types of foods. And also an improvement of if you want to have the right kind of breed that can be able to meet your goals, maybe in terms of milk and meat. So we see a lot of, impact that the livestock biotechnologies can have in these 4 areas. Next. And now with the synergies with the AU initiatives, actually, there's a very strong link with the CAADP. As I said, CAADP was conceptualized in the year 2023. And then when you look at the latest CAADP report, actually, no country was on course to meet those, those goals that were set in 2003 in Malabo. So this has necessitated actually, or made it possible to come up with a new strategy now, 2026 to 2035, that was recently endorsed in Kampala. And then there's also a very strong linkage with the science, technology, and innovation strategy for Africa. So which is the STISA, which expired in 2024. So, but right now we have STISA 2034, the strategy is in place. And at the moment, we are trying to draft the implementation plan, of course, in consultation with our partners at the national level and also at the regional economic communities level. So we also see a lot of synergies with the African Continental Free Trade Area, so which at the moment has 54 signatories, 28 have ratified, 27 have already signed, with the exception of Eritrea, of course. Next. And then these are just examples of the livestock biotechnologies you see across Africa, which are being done by Africa, wherever they are in the continent. And I think I'm happy to see that some of them are in the University of Edinburgh, ILRI, and others. And it's also important to note that in this, the three images that are on our screen, actually they're applying genome editing, which is one of the most efficient technologies that you can use to be able to shape responses to emerging challenges also in the livestock sector. And then other examples also you can see in the African ecosystem is there is a lot of examples where genomics approaches have been used to be able to identify best breeds, in Kenyan, Senegalese dairy cattle, Ethiopian sheep, and also to identify genetic variants which regulate resistance to diseases, among many other examples. Next. And then how do we sustain impact now with the livestock biotechnologies? In our view, I think it's very important to invest in skill development and infrastructure, because when you look at especially the Africa Innovation Report, which was recently released, you see that there's some parts of Africa which have fantastic infrastructure in place. Can you bring it back? The slides, please. The slides are gone. They're not on my screen. Please bring them back. Hello? Yeah, thank you, thank you. So I was talking about the need to invest in skills, development, infrastructure. We need to have more, strong public-private sector partnerships. We need to, advance our strategic communication and advocacy activities. We also need to look at, what sort of policies do we have in place? How do we harmonize them? And then how do we link them with the local, regional, and also the global value chains? And then lastly, we need to look at, livestock biotechnology as an innovation. I think with that, when we look at it as an innovation, then we can be able to, employ strategies that are able to support using, and also the interactions that also take place. So as I conclude, from the AU level, then I think we recognize that the sector is actually very critical for building resilience in the agri-food systems. And then it's also important for solving emerging challenges, especially in the era of climate change. And then, we're willing to partner with like-minded organizations to develop, review policies, enhance skills, create conducive environment, so that we continue to support realization of continental initiatives like CAADP and also STISA. So thank you for listening to me. Over.
Thank you. Christian, you gave us a marathon. 7 presentations in a very short time. Thank you very much. I think we'll now move to the last session, which is really a very brief discussion by some presenters. We need them to highlight one issue, only one issue. So I'd like to request the director of AU IBA, Dr. Huyam Saleh. Okay, and the director from CALRO, Dr. Martin Mwangi. Please come forward to sit on the hot seat here. And Professor Mizek Chagunda. Okay, I'll step down from here and come down. Thank you. Let's appreciate our 3 distinguished guests here. I have a very difficult task. And the task is to request our presenters to give a very brief answer, because I have actually, for the time given, I have 1 minute, and I'm requesting the audience, please, could I beg for 15 minutes of your time maximum? Because it's not only these 3 who have to speak. There are other 4 participants in the audience. I have Dr. Dr. Ruth Okong. Where is Ruth? Yes, please, if you could come and sit on the front row so that we can get your response easier. Then I have Dr. Paul Chege. Where is Dr. Paul Chege? Yes, sir, I've seen you. Yeah, you just join here. Ruth, Dr. Paul, please come and join. And then I have Dr. Nasir Yao. And finally, Dr. Nelson Ujango. Do I have enough seats for everybody? Let's appreciate everybody who's come to the front row. Now, if we were to divide the time ideally, we have 7, and I've requested for 15 minutes. So it's like 2 minutes maximum in response from everybody. Okay, and I was given questions, which I'm going to take the liberty to reduce the questions and decide on what I will ask. Now, if you notice the way I did, all the people who gave presentations were men. So, Madam, you're the first woman representative we have on the African Animal Breeders Network platform. So, you are our flag bearer. So I'm going to ask you, Africa has been missing the train of various historic revolutions, particularly because they left the women behind, including the knowledge and technology-driven transformation of the livestock industry. From technology adoption, transfer, and development, what should be different in Agenda 2023 and CAADP Action Plan? What do you feel should be different?
Thank you so much. First of all, let me say that Agenda 2063 has been developed with many inspiration, reflecting on adopting those biotechnologies. The same for the CAADP, and if we manage to look at the biennial reviews, we will see that the problems is coming from the AU member states are not funding agriculture in general, and in particular biotechnologies and innovations and research. And we need to address this issue through innovative ways and solutions. So for the CAADPs, you will find within the strategic objectives, there is many strategic objectives requesting member states to adopt innovations to increase production and productivities. But unfortunately, the communication and advocacy for this is so weak. There is no regulatories within our member states supporting the adoption and the usage of those innovations. And for that, we need to bring more entrepreneurship and investments in formalization of biotechnologies and tools. And here I'm not speaking about sophisticated tools, even the simple one. And also supporting and covering the gaps in extension for our farmers and pastoralists. Within the African Union Inter-African Bureau for Animal Resources, as mandated institution, specialized technical office for the African Union to lead and coordinate the livestock and the animal resources development in the continent, we are working with different partners, including CGIAR institutions and also the member states and the research institutions. For the genetics, many innovations has been done, but it needs more scale up, and that means needing budget. And considering the cuts in the developmental budgets, we need to think about public-private partnership, bringing more companies to support those pastoralists and farmers. And to ensure that we have promoting good governance and regulatory and monitoring those research and make sure that we are applying what those researchers are finding. Thank you.
Thank you. Thank you very much. I'd ask Dr. Yao. Is Dr. Yao here? Yes, please. Could you give your impression of what you think could be different in the Agenda 2063, having listened to what Madame has shared with us?
Yeah, thanks for giving me the floor. For me, what could be different is, I'm going straight forward to the point, by the way. What could be different is our way of doing things. Because when we talk about making change, how really do we make change? What are the key aspects, or what are the kind of mechanism that we should do differently, for example, to make a great impact really on the ground? For when we want to make a change in terms of implementation, okay, we need a kind of critical mass. And what are really the person that we really need to make the change, to make this change happen? Through the presentation, there are so many nice technology that has been developed. Fine, but how to use those kind of technology to really make impact on the ground? Who are the person to do it? What are the kind of critical mass that we should reach to get to those kind of things? And when I talk about critical mass, it's about human capacity and also infrastructure.
Okay, thank you. We've heard around the policy issues and around capacity. Dr. Martin from Calro, what policy options do you really think African governments can pursue?
Thank you. And my name is Martin Mwirigi, not Mwangi. Thank you for this opportunity. And let me say the Agenda 2063 is well crafted. However, I think the main challenge we have had is the implementation. Because as Dr. Atemo said, we don't have countries, especially that when we look at the analysis from the comprehensive Africa agricultural development, that aspect was not achieved. So basically what it means is we have either policies that are not helping in terms of them to be accomplished, or we don't have a framework that is being implemented or nationalized. So in terms of policies, there is an aspect of trade which is very key within Africa, and we need to be able to harmonize our legal framework, especially when it comes to Africa Free Trade Area Agreement. And that is where we need to be able to build access to products, especially the livestock products, so that you can have better movement. The other aspect has to do with support, especially for research, which is not there. Probably what is needed is to have a good policy guiding the way the livestock systems needs to be supported, especially at the research and training level. Because what is observed is you have institutions training on agriculture and especially on livestock. You go to all vet schools, they'll have an aspect, let's say, of embryo transfer. But I can tell you they teach theory. This is Africa. When you ask those even professors if actually they practice, they don't. All they do is teach. So we need to be able to have an arena where research is well supported, basically through policy that helps the financing aspect. Thank you.
Thank you. Thank you very much. We seem to keep gravitating towards capacity. It's like we go a little bit, but we keep coming back towards capacity. Is it Dr. Ruth? What particular policy options do you think must be pursued so that we can support the livestock sectors?
Yes. As a regulator, I believe for the African region, we need to come up with a harmonization, just like Dr. Murigi says, if we can harmonize our policies, because I think largely, apart from the capacity, we have issues with the acceptability of the new technologies. We can have all this running, but as long as we do not have enough public awareness to ensure that this wheel rolls so that the technologies that are developed are adopted, then we have a problem. We also have issues with political goodwill across Africa. Most of our African leaders don't per se support the new technologies. While we are in dire need of improved livestock, productivity, animals that are resistant to diseases, we need vaccines that are possibly GM-based or even gene-edited, we are having that challenge because once we have that support, we'll be able to move. Another area is funding. I think our governments hardly fund research. So could we have a policy that at least allocates a good amount of money into research and supports it? Then we'll be able to move forward to be able to achieve the agenda. Thank you.
Thank you very much. You have preempted my next question, which goes to Dr. Paul Chege, right, which was really, which approaches can we use to attract this funding? You know, how can we pursue or mobilize money or resources that we could invest in these areas? Can you give one or two, just one, give me one example of what we can do?
Julie, you're asking the question I wasn't prepared for, but that's okay. I will tackle it anyway. My name is Paul Chege. I work for STISA AfriCenter, and we communicate on science. And what has been spoken here by the earlier speakers and panelists is very exciting, I would say, and it's an opportunity for learning all the nice things that we are doing. But there are several things that have been mentioned that are key. One is data exchange. I would think that, you know, working in silos will not help us. We need to, you know, and I liked what Professor Raphael kept repeating, multi-country approaches, that will help us. Another thing is on awareness creation, and I would like to draw the attention of all listeners on the deliberate war on science. We could do all great things, but then if we are not aware of the war that is being waged upon the technologies that are coming, then we won't make a lot of progress. And being in Erie, you know, we are a hosted institution in Erie campus, and we see all the good things that Erie does, especially development of vaccines. And recently, Kenya government tried to roll out vaccines, and you saw the eruption that was there. And what caused all this, I think it's misinformation. We have to be aware that the carriers of the messages these days, messages on science, have changed. There's growing mistrust on the conventional sources of information. There is growing trust on, you know, what we call experts on social media, quote unquote experts, without really verified facts. So we need to support initiatives that you know, consider communication aspects in all initiatives that we do.
Thank you very much. So you brought in the aspect of communication, and the way in which messages are communicated. I'll go to Professor Mizek. How can the benefits of technology be harnessed for growth and productivity while ensuring that we are actually narrowing the gap? We have a gap in technology adoption, we have a gap in the knowledge between the North and the South. How can we harness the benefits of technology?
How can we harness the benefits of technology? So this afternoon, the presentations, we had 7 of them, are actually a very good demonstration on how we can harness the benefits of technology. We started with a scientific presentation from Tom, and then we moved on to another scientific presentation coming from Raphael. That's research and development activities. Then we actually had ABNet coming in, which is a society of practitioners. We moved on to a university-based spin-off, COILIB. And then we moved on to African Union in the framework. To me, that's a real good demonstration on a pipeline on how science can move from the lab to the plate. How science can move from the lab into impact. The way we should harness the impact of science or the contribution of science is to work in partnership and not to say, that's for scientists, that's for Tom who works with cells in the lab. No, that technology can have its impact if we actually understand what we are doing. So it's a combination of science, research, and training. Not only to build capacity, but also capabilities. Thank you.
Thank you very much. And I'll throw a similar question to Dr. Nelson, which is how can the benefits of technology be harnessed for growth and productivity? Now, hopefully, these are very, very scientific angles. We are hoping to look more from the producer side. How can they benefit from technology for growth and productivity in the producer sector?
Thank you, Julie. My name is Nelson Ojango. I come to you from Kenya Livestock Breeders Association. We have been lucky to be in existence for a bit. We run the Kenya Stat Book and the Dairy Recording Services of Kenya, so that's what we do. How can this, there's been lots of presentations today, this evening, and I think from the farmer's point of view, we would like to see solutions on the ground. We'd like to see solutions on the ground, and the issues that itch us, the issues that worry us, the issues that are important to us are to do with productivity, and that has been addressed. They have to do with disease, that has also been addressed. Climate smart solutions to livestock production, that has also been addressed. I'd like to add something else, disease diagnosis, biotechnology for disease diagnosis, that would be useful. And these solutions should have relevance, they should be affordable, cost-effective, they should be implementable, they should be practical, accessible, and disseminable through appropriate channels. Maybe I should stop there.
Accessible, disseminable. Mizek, I come back to you as the man in charge of CTLGH. Are they accessible?
I would say yes, they are accessible. So at CTLGH, we work in partnership and we make sure that the science that is developed in CTLGH is passed on to what we call the farmer-facing organizations or initiatives. And that's where all these technologies really become accessible to the end users.
Thank you. I'd like to give the last word to our director from AUIBA, in how does she see all these different technologies being accessible at the different levels across countries and across systems.
Yeah, thank you. I think to do that, we need, first of all, to ensure that all the policies and all these biotechnological tools to be domesticated at the national level at the EU member states. I think this is very important. And to do that, we need resources, and we need also the interest from the public sector and the private sector at this national level. And I think as an African Union institution with the different partners, we are able to do that through different projects. Many piloting projects has been implemented, and there is buy-in from the producers. What we need is to continue pushing for the adoption of these tools by the pastoral communities and farmers at the country level. And to do that, we need resources, of course, and we need commitment from the African Union member states to make sure that they are increasing the fund for the livestock sector in their countries.
Thank you very much. I think that is all we have time to hear. We would have loved to hear a lot more, but at the end of the day, we want to say thank you to all of you for giving us your time. The questions I know were not planned, right? But it's nice to hear fresh thoughts, a different perspective. And I'm thankful to all of you for giving us your time. Let's appreciate our panel members. Thank you. And as we close the evening, I'd like to ask Professor Mizek to give a vote of thanks.
Thank you very much. So, to start with, as Director of the Center for Tropical Livestock Genetics and Health, CTLGH, I should say a very, very big thank you to you all for coming to this session, which was starting sometime late in the afternoon, when some of you were already planning to go and have a taste of nyama choma in the city, but you actually decided to come here and have a taste of science. So thank you very much for being here. I would like to thank the organizers. So as director of CTLGH, I did not actually organize this. I had a team led by Dr. Christian Tiambo. I would like you, ladies and gentlemen, to join me thanking the team that organized this session. Let me also really thank the presenters very much for their presentations and really their thought-provoking questions and insights. Ladies and gentlemen, let's give an applause to all the presenters. The panelists sitting in front here have also shared with us a lot of good insights. Thank you very much. Join me, let's give them an applause. Obviously, last but not the least in importance, I would like to really sincerely thank Dr. Julie Ojango, who has moderated this session very professionally. Thank you, Julie. And indeed, at the end of it, let's thank each and every one of us. So let's clap hands for ourselves as participants of this session. I hope it's not only me who is going away thinking I've learned something, there is something to be changed. I hope you've also learned one or two things, and I hope we can still be partners in science for the betterment of the smallholder farmers, and at the end of the day, for the betterment of Africa as we contribute to the Africa we want. Thank you very much. I'm told there's a reception, and this reception is upstairs. There is somebody who will stand by the door and show us the way to go. I'm told there are some drinks. At the same time, I was told to emphasize that there's some fine wine, which will take away the dust from your throats after swallowing all the science. Thank you very much.